everyone. Questions before we get started? All right, let's get started. We're going to finish up hormones hopefully today so we can start into steroids next week. And then, so steroids, like anabolic, androgenic steroids, the performance enhancing, not just hormones made from cholesterol. Like that's steroids, fine. But we're not talking about aldosterone. You don't have professional athletes doing aldosterone and prednisone and like, oh, I just need more cortisol. I just need to be super stressed. So anabolic, androgenic steroids, testosterone, a bunch of exogenous administration of these things. That's next week. And and then we'll talk about enzymes, which is a lot of cell signaling cascades, phosphorylation cascades. Often they'll just call it what it is, phosphorylation cascade. And that leads us into mTOR. And so maybe we'll do one lecture on, all right, let's talk about hypertrophy in a very general sense, and then let's get into mTOR and how all of that works. Then we'll do supplements that interact. And there are some supplements that work. There are some that actually are legitimately effective, and they're not necessarily the ones that everyone's buying. So we know this stuff, neuroendocrine system. We know how it is connected, the hypothalamic, pituitary, whatever axis. And so if I ask a question, what regulates X? Warren Beatty, right? If it's that 1981 version of Trivial Pursuit, the genus edition, the first edition, hypothalamus regulates everything. It's the brain region. It's not a gland. It's a brain region, and it does all of your regulation, your control, your regulation. It's a CEO. Remember, we had that office example. The CEO, the owner, CEO. That's your hypothalamus, and then it relays its messages. Neuroendocrine history. It didn't begin in the brain, right? It began in the testes, 1849. Arnold Berthold took it was chickens and their testes and castration, transplantation, and swapping, the creepiest one. And Oscar Minkowski was 40 years later with a dog pancreas, right? Just take the pancreas out of a dog and see what happens. And there's global effects, right? It's all over the place. This is horrible. And so there are these glands, there are these hunks of meat, of tissue throughout the body that have global effects, endocrine effects, global effects. And so the only one you really need to know about is the first one is the Arnold Berthold and the chicken, sort of the foundation of this understanding that there are these chemicals released by glands into the blood. That blood goes all over the place, it circulates everywhere. And so those effects of those little compounds, the effects of those hormones are systemic. Now, the hypothalamic pituitary, whatever axis, from the hypothalamus to the anterior pituitary, that's blood. It gets there by a capillary network. It doesn't go into systemic circulation, right? It gets there by way of blood. This portal system, just a straight shot, a road that goes from one to the next. And the anterior pituitary is where you're releasing almost all of everything that you really, this is the governing hormones. The posterior pituitary, this is nerve, it's connected by nerves. The cell bodies are upstairs and the terminals are downstairs in the pituitary. And so oxytocin and vasopressin, that's it. That's the stuff that's coming out of your posterior pituitary. So vasopressin is, peeing stuff. It's antidiuretic hormone, arginine, vasopressin. Keep your pee in. Oxytocin is like, let's get into expulsion mode with babies, and dilate the cervix and contractions and whatever, and let's get a nice cozy effect after cuddling. So that's your oxytocin. But so that's the posterior nerves. Anterior is blood. Again, just these axes of the adrenal gland is the, I don't know, bad wig or toupee or like hideous hat at the top of the kidneys. There's the adrenal medulla, and the cortex. The cortex are steroid hormones, and the medulla, that's going to be your non-steroid hormone. Your epi is coming from the adrenal medulla, and cortisol from the cortex. But hypothalamic pituitary thyroid, hypothalamic pituitary gonadal, hypothalamic pituitary liver. And so we'll talk about growth hormone today a little bit more as we move into uh, hypertrophy. So testosterone and growth hormone is some of these things that are really doing it. Hormones, these things are endocrine hormones are travel a long ways. Autocrine doesn't travel far at all. That's just the cell that releases it is the cell that benefits from it, or maybe it's not a benefit, maybe you don't want it affecting that cell, that is tormented by itself. But so that is autocrine, paracrine, right, adjacent, but for the most part, long range communication. And neurotransmitters, very short range. Neurotransmitters, very short range and very fast and all or none, where hormones tend to be concentration based. How much insulin is there? You don't get a lot of insulin in a needle and you're going to go into a diabetic coma. A little bit of insulin isn't going to have much of an effect. A ton of testosterone and you look like Ronnie Coleman. A little bit of testosterone boost and it doesn't do anything. So there are concentration. That doesn't do anything, but when people are doing research on testosterone, they're not usually doing Ronnie Coleman doses. They're doing therapeutic, if you have muscle wasting, if you're cachectic, you have HIV or cancer related muscle wasting, and maybe we give them a little bit of extra testosterone, something like that. We'll talk about that stuff soon, but concentration based. So the two classes of hormones, let's ignore icosanoids, the two classes of hormones, and you know what icosanoids are because we've been talking about it, like prostaglandin, stuff from arachidonic acid. PLA2 releases your arachidonic acid from your cell walls, from your phospholipid bilayers, and then it gets converted into all your icosanoids. And sure, fine, hormonal-ish. Call it a hormone if you want. Call it just icosanoids and sort of, I don't know, immune and kind of chemical interactions with tissues if you want. Just depends on your definition of hormone. Hormone is sort of a vague word. So we have stuff made from amino acids over here, this sort of gray, blue, cloudy sky colored, the amine, the peptide, the protein hormones. And so like, norepi, epi, or dopamine, let's get all the catecholamines in there, but epinephrine is really the one we're going to talk about. And made from tyrosine. That's just tyrosine modified a bunch, and not that much, but a modification of tyrosine, the amino acid, and now you have your epinephrine or your dopamine or norepi or whatever, L-dopa, whatever. So an amine hormone, it's just an amino acid, right? A peptide. Let's string together a bunch of amino acids, and a protein hormone. So there's oxytocin. This is let's go cuddle, and a protein hormone, growth hormone. There's 191 amino acids in that sort of canonical version of growth hormone, and steroid hormones totally different. Steroid hormones, this isn't just a bunch of linked amino acids, it's cholesterol. That's cholesterol. And it's you make modifications of cholesterol. You don't just link together amino acids and maybe make a little modification here and there. It's cholesterol. And so whether it's estrogens or androgens or anything from the gonads, right, ovaries and testes, or anything from the adrenal cortex, that's going to be your steroid hormones. We'll talk a little bit more about those, but again, made from cholesterol, they're fat soluble. So that's what allows them to diffuse across the sarcolemma. Peptides are not fat soluble. They're water soluble. They can't diffuse across. And where they're made, peptides, these things are made all over the place, just cells. But there's a ton of glands that are making them, but these things can be made in a lot of areas. Steroid hormones are this is really your hubs for where those are coming from. The nucleus, right, nuclear or cytosolic, sort of outside of the nucleus within the cell. Receptors, that's where those receptors are. They aren't cell surface receptors for steroids. For peptides, they are cell surface receptors with, T3, T4, the exception. We'll talk a little more about that today. The thyroid, peptide hormones, amine hormones from the thyroid. But steroid slow initiation. The steroid hormones take time. Genomic effects are sluggish. Genomic effects, if you're going to do some genome thing, this takes time. And so if you want to know what's happening here, oh, there's some hormone and what its effect is, and it's just taken forever to see if it's like an effect on blood sugar or something, or it's an effect on lipolysis, let's say. You're doing some effect on lipolysis, and it's taken forever. It's taken like hours for this thing. Tumor necrosis factor, TNF, TNF alpha, TNF. Sometimes this just takes a long time. So there's an indication there's genomic effects happening. Non-genomic stuff, that's hasty, that's fast. And that's stuff like GLUT4 translocation or your heart contractility, all of these non-genomic things. These are fast, and you can see how fast they happen. But cortisol versus epinephrine, that thing is fast. Epinephrine's very fast. Cortisol is slow. It has similar effects, but it's very sluggish. And when you start getting into steroid hormones, they tend to be longer acting with a lot of nuclear receptors, and peptides are just these quick, cell signaling cascades, very fast effects. There's genomic stuff, but peptides tend to be faster. So if we're going to say 3 classes, if we're going to say 4 classes, fine, let's throw in icosanoids, but they behave just like this, right? They have these cell surface receptors, and prostaglandin signaling, and we're going to talk more about that soon as we get into mTOR and MAPK, mitogen activated protein kinase, and some of these signaling cascades. We'll talk about prostaglandins. It's not just ow, 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 a marker of damage. It's also an instigator of repair. So icosanoids are they are released proportionate to tissue damage, and delayed onset muscle soreness. Ow, it hurts when I'm doing this. That's letting you know that there's a bunch of tissue damage, but it's also signaling repair. It's not just waving the flag and saying, okay, it's over here, the injury's over here. It's also binding to prostaglandin receptors. Yeah, let's cause some pain, but let's also repair things. So icosanoids are going to behave like these polypeptide classes of hormones. So epinephrine, it's an amine hormone, right? The adrenal medulla. Adrenergic receptors, beta adrenergic receptors are what we talk about. Beta 1s, this is mostly cardiac tissue. Beta 2s, and I'm not going to ask this level of detail on the test, but beta 2s, you're looking at muscle, you're looking at liver, those tremors, right? If somebody is taking clenbuterol, it's a drug for bodybuilders, and especially like a cut phase drug for human performance. Clenbuterol is this oral beta 2 agonist. That's kind of muscle, that's liver mobilized carbs, stuff like that. Beta 3s, these are your lipolysis, that's your fat. We're not going to talk about the alphas. Nervous system, or nerves, neurons, right? Their brain, there's some of the kidneys. We're not going to get into the alphas. The betas are what we care about. And so beta adrenergic receptor is what I keep saying. Beta adrenergic receptor. And if we're in fat, we're talking beta 3s, right? If we're looking at maybe the liver, some glycogenolysis, we're looking at beta 2s. But beta adrenergic receptor, let's lump them all into the same group. But so these, tyrosine. I mean, it's the same thing here, actually, tyrosine. But this has to bind to a cell surface receptor, no matter what it is. It could be insulin, it could be growth hormone, it could be glucagon, whatever it is, polypeptide class of hormones. Cell surface receptors, they bind to a cell surface and relay their message inside. Testosterone is a steroid hormone, right? You see the cholesterol right there, and mostly testes, yeah, sure, a little adrenal, but mostly testes. And androgen receptors, we're inside of the cell here. So it goes through the, let's say it's muscle, sarcolemma. We're going through that cell surface membrane, that phospholipid bilayer, and it binds in the cytoplasm. It translocates to the nucleus, and then it regulates gene expression. Very slow. Testosterone is not fast. This is a slow guy, but it's long lasting, right? You find somebody on steroids and they're doing their injections once a week, something like that. It lasts a long time, but it's slow acting. You do your testosterone on day 1. They'd be like, I'm huge on day 3. That's not how it works. Triiodothyronine, that's T3. Thyroxine is T4, but T3 and T4 are your two thyroid hormones, and same amino acid or amine hormones. The receptor, they have to cross the membrane, right? They cross the membrane via these transporters. I'll talk a little bit about that. They have genomic effects, a little bit slower stuff, but non-genomic effects. They also have signaling cascade roles. When we're looking at PI3K, PKB, sort of canonical mTOR signaling, they're also working through that cascade. So these are a hybrid, although they are really similar in structure, right? To especially like epinephrine, they're kind of similar there. These are essentially this in structure, but they sort of behave like testosterone, because testosterone also, or like steroid hormones, testosterone also has these secondary effects of cell signaling cascades, and we'll talk about those later, into next week at the earliest, probably. We'll talk about that. Triiodothyronine, that's more potent than T4. Really, what the thyroid is releasing more of is T4, but T3 is a little bit more, has a little bit more interaction with the tissues. And so, that is it's a hybrid role. So let's go through some of these individually, right? Epinephrine. So we know there's a beta adrenergic receptor. It's in fat, right? We're seeing hormone sensitive lipase and perilipin. That's what this guy is. His jacket has now become hunter green. I feel like that's a little bit creepier than brown. And so that would be perilipin, right? When PKA, protein kinase A, phosphorylase perilipin, we're exposing on those triglycerides, triacyl glycerol or triglyceride, a glycerol with three fats on it, strings of fatty acid strings on it. And you have three of those strings of fatty acids on a glycerol. So this, we're talking beta 3s, right? Beta 3s, that's going to be adipose tissue. That's going to be lipolysis. That's your beta 3s. But who cares? A beta adrenergic receptor. That's all it says. It doesn't tell you which class it is. And this is not like levers where I care about class. What's a lever? It's a lever. That's all you need to do for beta adrenergic receptor. We're activating adenylate cyclase. And adenylate cyclase is going to convert ATP into cyclic AMP. Cyclic AMP binds to PKA. PKA really should be the big thing here. The big, like, this is it. This is the thing that does it. The big regulatory enzyme, PKA, protein kinase A. And it phosphorylates tons of stuff, right? Hormone sensitive lipase and perilipin. It's phosphorylating lots of stuff. Now, technically, it says hormone sensitive lipase is the first one. Adipose triglyceride lipase is technically the first one. It's a little bit different from this cascade, but who cares? I'm not going to ask you that on the test. So PKA phosphorylates a bunch of stuff, hormone sensitive lipase and perilipin, and really gets this, especially perilipin. You're not going to go through lipolysis without opening up and exposing the triacylglycerides or triglyceride, triacylglycerol, without exposing those things. So that's the signal. And you can see just tyrosine. You can buy tyrosine as just a amino acid. I have it. I put it in like what if I make some sort of shake of some sort in my coffee, something like that, I'll dump a little bit of tyrosine into it. But now we have our catecholamines. So if you want to know what the catecholamines are that I refer to, all right, there's your catecholamines, right? Epinephrine is really the one I'm going to talk about. Norepi, that's neurotransmitter version of it, almost entirely. Epinephrine is your hormone version of endocrine version. Dump it into the blood and let it do its business. Now, again, triiodothyronine. That's T3. That is the more potent of the thyroid hormones, T3, T4. And the way it functions, it has a bunch of results, but skeletal, this is a muscle physiology class. I'm not talking about the thyroid for fun, right? Just because, like, well, it's a hormone and exercise physiology. Well, it's actually muscle. It's really important to muscle regeneration. I mean, incredibly important. And so this is a study on like rats and ferrets and stuff. But when you look at the thyroid transporters, these are both transporters, these two. And these transporters will escort the thyroid hormones across the cell surface, get it into the interior. And if you get KO, knockout, right? So wild type, so this is just like you're a rat or a ferret. I don't know what animal this particular one. you knock out one of the transporters, and MyoD, you remember MyoD, we just talked about that a week ago, a couple weeks ago, something like that, when we were talking about myositis ossificans and fibroblast differentiation, and we want our fibroblasts in muscle not to be laying down bone matrix. So MyoD is responsible. So in recovery, you injure yourself, and this recovery fibroblast influx and differentiation, and let's repair all of this tissue. MyoD is critical for that. When you see this knockout of this transporter, you get a reduction of MyoD. Same thing with this transporter, a reduction of MyoD. Knock them both out. That's what this last column is. Knock both of those out. And that MyoD, myoblast determination protein, MyoD, it goes way down when you knock out both of these things. And so you're looking at the thyroid being critical for muscle regeneration. And again, that's the more important one. Happens if you have hypothyroidism. I haven't seen the literature on hypothyroidism and what happens to muscle regeneration in exercise, but my hunch is it's really compromised. And I'll talk about the mechanisms. I mean, there are genomic effects, transcription factor sorts of effects, but when you look at the thyroid's effect, and we'll talk about this a little bit today on PI3K, PKB, mTOR signaling. Let's translate proteins. Let's create a bunch of proteins. Then hypothyroidism, I can't imagine a situation in which an inadequately functioning thyroid is going to be compensated by other mechanisms. There's a ton of overlap in the body, tons of overlap in the body, but you want them to be overlapping, right? You don't want one system to be overburdened because it's probably not going to pull all the weight that of the team members that have left. So I don't have a perfect answer for that, but my answer you can rely on with some confidence is that muscle regeneration, repair, remodeling, right? Your recuperative power between workouts and your ability to go exercise on Monday, get back on the field or court or diamond if it's baseball on Tuesday or Wednesday or whatever day I said first, that's going to be impaired. Your ability to generate new muscle to hypertrophy is going to be impaired. Now, I can't tell you how much, what the deficit looks like. I haven't seen literature on how to quantify the deficit, how much impairment happens. I'm not sure. What you said in the text box, compromised recovery. Yeah, compromised recovery, but also compromised progress, because progress is a form of recovery. Autocrine, paracrine, endocrine signaling, different types of hormones go in different ways. Now, most hormones we think of as endocrine, but paracrine, again, adjacent tissues, a lot of growth factors, clotting factors, growth factors. These are adjacent tissues. And you don't necessarily want, when you're having disruption of some particular tissue, you're doing a calf workout or something, you're not necessarily going to have all of these clotting factors and stuff up in your biceps. I mean, unless it's calves and hamstring biceps. But autocrine, vascular endothelial growth factor, the mechanogrowth factor, it's IGF. This is basically IGF, but it's a version of it. It's in the skeletal muscle. And some immune stuff, interleukins, some immune stuff. Myostatin is autocrine. Myostatin is paracrine. Myostatin is endocrine. So some of this stuff has roles all over the place. You could say the same thing about IGF. You could put IGF in all of them, but we're going to talk more about myostatins as inhibitory myokines, a little protein, think a little cytokine signaling proteins, a little protein myokine, muscle myokine, and it inhibits skeletal muscle growth, and we'll talk about it more. So getting into cell signaling cascades, remember this guy, Earl Sutherland, he first discovered cyclic AMP. And any cyclic GMP it can be your second messenger molecule, guanine triphosphate, supposed to adenosine. So, but whatever, cyclic AMP first discovered, and this really is this is incredibly important second messenger molecule. You have something happen at the cell surface. You're over here at the liver. This is going to be more glucagon, right? Epinephrine is going to be more Muscle is a little bit is more sensitive to epinephrine, but we're going to mobilize carbohydrates. Like, let's break up glycogenolysis. Let's break up our stored carbohydrates and get them on the march. Let's march them to the oven. We're going to burn these things, but you can't burn a whole huge hunk of glycogen, right? You have to chop off little chunks. It's G1P. We'll talk about that later. G1P, G6P, what that is versus glucose, right? But this is the glucose-y chunk where the phosphate is. Doesn't matter. So adenylate cyclase takes ATP, breaks it down to cyclic AMP. Cyclic AMP binds to protein kinase A, binds some of those, and you're going to turn it on, right? So you're going to turn this on. Now, protein kinase A phosphorylates stuff. It initiates, well, it's both downstream and upstream from and to phosphorylation cascades. If you're going to phosphorylate a bunch of stuff, and that stuff is catabolic, protein kinase A is one of your go-to enzymes. Kinase phosphorylate stuff. It phosphorylates down stuff. So for carbohydrates, phosphorylase kinase, it turns that on. It turns off glycogen synthase because it phosphorylates it, but that turns it off because you don't want to put carbs back together with glycogen synthase. You don't want to put carbs back together while you're trying to break them apart. That's silly, right? You want to stop depositing money in the bank when you're withdrawing money. So glycogen phosphorylase, when you turn that on, you're withdrawing carbohydrates from your bank, from your glycogen bank. You're withdrawing usable carbohydrates, dollars from your bank account. And this would be putting money back in. You're not going to be doing the same at the same time. That's stupid, right? So you turn off deposits and you turn on withdrawals. And so that is glycogenolysis. Now, lipolysis, lipid lysis, right? Let's chop up fats. Let's take some triglycerides and lyse them. Lipid lysis, lipolysis. We've talked about epinephrine, glucagon. Here you see atrial natriuretic peptide. Fine, right? That goes through guanine. Where are we here? There. As opposed to cyclic AMP, we have cyclic GMP, but that hormone sensitive lipase and perilipin, it's doing the exact same things. You see adipose triglyceride lipase here, so this diagram gets it right. But so fine, atrial natriuretic peptide, let's mobilize some fats. Fine, yeah, let's add that one to the list. Okay. Catecholamines, right? Epinephrine, norepinephrine, adenylate cyclase, CAMP, CAMP, cyclic AMP, adenosine monophosphate as opposed to guanine monophosphate. PKG, protein kinase G, who cares? You don't need to know PKG, like smooth muscle stuff. It's another kinase, and we talk about PKA and PKB because PKA is super catabolic, PKB is super anabolic, and muscle metabolism, are we growing or are we shrinking? Is it hypertrophy or atrophy? A lot of this is a balance between PKA and PKB signaling. What you turn on these things or turn them off. So we're not really going to talk about this one, but atrial natriuretic peptide, sure. Now, this is just an article about atrial natriuretic peptide and how it works too. Now, growth hormone, sort of the least important on the list. It's not the least understood because it seems to just amplify other stuff, right? It seems to amplify other effects, whether it's beta-adrenergic receptors or adenylate cyclase specifically, phosphodiesterase interaction. It just seems to be interacting with other things. It doesn't seem to have its own direct effect. Cortisol has its own direct effect. You bind to a glucocorticoid receptor. Let's see, where are we? There, GR, glucocorticoid receptor. Glucocorticoid, that's cortisol, right? Glucocorticoid. Bind to that, and for one, that's PDE3B, so this is the lipid version. There's tons of phosphodiesterases, different phosphodiesterase inhibitors. A lot of you have had a phosphodiesterase inhibitor today. You have had a non-specific phosphodiesterase inhibitor probably this morning. It probably came in a mug or in the form of chocolate. Is a different one. We'll talk about those soon when we do more cell signaling cascades and getting a little bit more into it. But obviously I'm talking about caffeine. The one everyone has all the time is a non-specific PDE inhibitor, but the glucocorticoid receptor, okay, we're inhibiting PDE, but we're also promoting cyclic AMP. So this would be what you see right here is an autocrine signal. Right there would be an autocrine signal increasing cyclic AMP. So we're increasing cyclic AMP through one mechanism, an autocrine mechanism, and we are inhibiting its breakdown by inhibiting PDE. So cortisol is mobilizing fat. It's not good for adiposity. It's not like, oh, I want as much cortisol as I can get, so I can do more lipolysis and burn more fat. Not really good. It's good for the transcription of some lipolytic enzymes, and let's get some machinery here that's good for lipolysis. Sure, it's good for that, but lipoprotein lipase LPL activity, it's really, you're just sort of shuffling around where your fat is stored. You just wind up with like a bunch of stomach fat. You don't want a bunch of cortisol, but the effect of cortisol on increasing lipolysis, it does have an effect of increasing lipolysis. This is exactly what you saw on the previous slide right here, right? And then its effects increasing cyclic AMP. So if you can reduce phosphodiesterase, again 3B specifically, that's the fat one. Increase cyclic AMP, you are increasing the activation of PKA, protein kinase A, phosphorylating hormone-sensitive lipase, phosphorylating perilipin, and getting your triglycerides broken down. So you have a bunch of fatty acids and glycerol, that 3-carbon carrier molecule. So tumor necrosis factor, this is the most complicated of all of them. It has so many interactions, and some good, some bad, a lot of bad. But there's just tumor necrosis, this little alpha, tumor necrosis factor or tumor necrosis factor alpha, fine, either one. And TNF, is usually how it is. You'll see it in the literature. In the most recent literature, if it's something 2020, they just call it TNF. But a lot of immune regulation is happening here. But you see this cascade, extracellular signal regulated kinase. That's what that is. That's MAPK signaling side, mitogen activated protein kinase. But AMPK, that's probably if PKA, I say it's like arguably the most catabolic enzyme in the body. AMPK is probably the most, but PKA is I would say it's runner up. Either of them, you can make a compelling case. What's the most catabolic enzyme in the body? Break stuff down, right? Catabolic, not hypertrophy, atrophy, right? Break down all of your energy substrates and get energy ready to be consumed. And AMPK and PKA are both. You can make a case for either of those. PKB, you could make a case for the most anabolic. mTOR, you can make a case for the most anabolic. That's another enzyme. Who cares? So TNF, though, it has a lot of effects. Some of them are genomic, and so some of that is going to be a little bit more sluggish. You can increase cyclic AMP with it, transcriptional regulation with it. I don't, it's that one's the most complicated. Tumor necrosis factor. Everything else is pretty well understood. Growth factor is just, actually, it's not that well understood. Tumor necrosis factor and human growth hormone, those interactions with lipolysis are a little bit more compound, right? Those are a little bit more complicated, and this one isn't that well understood. But there's a lot of mechanisms that have been tons of mechanisms, really well studied, has been identified. This just seems to work through other mechanisms. And this is just these, gene expression. If we're looking at transcriptional regulation, gene expression from TNF, that's what this diagram is showing. It actually is a really good article. If you wanted to read the article, TNF alpha and adipocyte biology, I mean, it came out over a decade ago, but it's really good. It's probably the best article I've ever seen on TNF and adipose. But yeah, the regulation, a bunch of signaling cascades, right? These TNF receptors and what they're doing. Tons of stuff, right? Now you don't need to know, the rest of it. I mean, like bile acids. There's lots of stuff that can affect lipolysis, that can affect the mobilization of fatty acids. And exercise is even exercise intensity matters. So maybe we'll talk about this at some point this semester. But people will say, oh, just increase at higher intensities. And I can make an argument where everyone should increase at higher and higher and higher intensities because that's the threat. You could make that same argument. That's the threat is a higher intensity. Casual walks are metabolic, but it's not cardio. Don't call it, oh, I'm doing my cardio. You mean you're walking and your heart is totally unthreatened? It's beating like 85 beats a minute. Like that's not going to give you a heart attack, right? There's no infarction that's going to result from your casual walk on the treadmill, or that's not cardio. It's metabo, right? You're doing metabolism. That's all you're doing is you're burning some extra calories. Now, what people will say is you're increasing your calories, increasing your calories, increasing your calories. Your MET value, metabolic equivalent of the task. And whether you're 2 times your resting metabolism, 3 times your resting metabolism, 4 times your resting metabolism, 5 times your resting metabolism, that's 2, 3, 4, 5 METs, right? The higher the MET value, the more threatening it is to your physiology, to your anatomy, what are your bones and your muscles and whatever sinewy things and glands and organs. It gets more and more threatening the higher intensity. And what is our body responding to? Protection, survival, right? I want to go on living for as long as I can. So in the interest of self-preservation, I'm going to reinforce my heart and blood vessels and all of these tissues to be able to withstand higher intensity. So high intensity cardio, yeah, it's better, but you get too high intensity and you actually burn less fat than if you back off on the intensity, because maybe you'll go through a bunch of lipolysis, but you're not really releasing the free fatty acids. So as you increase and increase and increase intensity, what people will say, you listen to the personal trainer or something, what they'll say is, you get to a certain threshold and you start consuming carbs more and more and more, and it becomes proportionately higher carbohydrate. True, totally true. Where you're just burning more and more calories with each additional MET you add to your performance. And you go from 10 METs to 11 METs, from 11 METs to 12 METs. You're getting pretty intense here, 10, 11, 12 times your resting metabolism. You're getting really intense. And you get to a point, though, where you actually consume even less fat as you add another MET. It's not like, well, you've maxed out your fat metabolism. Fat metabolism can only go so fast. And from then on, it's just a bunch of carbs in addition to that. And so if you want to burn as much fat as you can, really max out the intensity, because you're going to burn maximum amount of fat and the carb. That's not actually how it works. And we can maybe talk about that later, but let's talk about insulin now. And so or norepi is epinephrine. Let's just say this is epinephrine. And so this is an amine hormone, adrenal medulla. It's released. It's going to bind. We're in fat, right? So we're looking at beta 3s. We're in fat. And it binds to a beta adrenergic receptor and initiates its signaling cascade, right? So we know how that PKA signaling works, adenylyl cyclase, stuff. But insulin is inhibiting it. Insulin inhibits it. But insulin does not inhibit this, right? The receptor, it doesn't go like bind to the receptor. It's not like competitive inhibition, which we'll talk about next week. We'll talk about competitive and non-competitive and uncompetitive, all these different forms of inhibition. We'll talk about that. That's not how insulin works. Insulin doesn't inhibit this enzyme. It breaks down cyclic AMP. It converts it into AMP, right? And so that's phosphodiesterase is what it's doing. So insulin receptor, obviously insulin binds to that. You can get IGF to bind to it, but insulin receptor substrate, you go through PI3K, phosphatidylinositol 3 kinase. That's a kinase, and eventually you get to PKB, AKT or PKB. Downstream from PKB, we're mobilizing GLUT4. Downstream from PKB is mobilization of GLUT4. And downstream from both PI3K and PKB is activation of PDE, phosphodiesterase. Now, a lot of these diagrams, if you Google stuff, you'll see a lot of diagrams. They're just different. These are 2 different diagrams. Obviously, right here is the middle of the screen where the diagrams are different because they're totally different colors, and the cell wall stops. But like insulin receptor, and PIP2 becomes PIP3. But what you see missing is PI3K. What converts PIP2 into PIP3? Phosphatidylinositol bisphosphate. You can say diphosphate or biphosphate. Who cares? Phosphatidylinositol bisphosphate, whatever, two phosphates to triphosphate PIP3. That's actually PI3K is what's doing that, and it's missing. You don't even see PI3K, or at least I don't, and maybe it's on here and I just don't see it. On this one, you just see insulin PI3K. This is the only thing you see. So these diagrams are different. There's tons of stuff on this one. There's missing one thing. This diagram is the only thing on there is the thing that is missing. And so the mobilization of your GLUT4, the mobilization of GLUT4 is going to be downstream from PKB, but PI3K and PKB both seem to be activating phosphodiesterase, and phosphodiesterase is converting cyclic AMP into AMP. Here's your regular AMP, right? This is your phosphate. your monophosphate, right? The one. And cyclic AMP, this is it right here. This is the cyclic portion is right here. That's the difference. Pretty much, it's almost the same thing. Now, what phosphodiesterase does is just go whoop and convert it into this. That's what phosphodiesterase does. This has been studied for a really long time. It is not new. Um, understanding of phosphodiesterase and its inhibitors and its function, not at all new. And we know there are, if we're looking at beta adrenergic, receptors, and there are, stimulators and there are inhibitors. When you look at like opioids and cannabinoid receptors can be inhibiting this cascade. Beta adrenergic receptors can be enhancing that cascade. But once you have your cyclic AMP made, the way you shut down that cascade is you can shut it down at any number of sites. A really good way to shut this down, to shut down signaling, is phosphodiesterase. Just to look at what the stuff is doing. This is a nice chapter. Phosphatidylinositol 3 kinase. Again, that has different names too. PI3K is always what you see it as, but sometimes you'll see it abbreviated. When they spell it out, sometimes they spell things out differently. Adenylyl cyclase, adenylyl cyclase. Again, who cares? A bunch of names for whatever. And this is just when we're looking at PDE, we see this article saying, you know what? It is not just PKB downstream from PI3K. It's not just PKB doing it. It seems to be both PI3K and PKB are knocking out, are activating PDE. So both PI3K and PKB are inhibiting. Because wart mannan, we'll talk about that more as we go forward into it's like introduced. It's a metabolite of this fungus, but it's a PI3K inhibitor, a very well used PI3K inhibitor. So if you just inhibit PKB, you can still get PDE activation. But if you introduce wart mannan, which blocks out off this, and this is downstream from this, so if you block this, you're also blocking this. You just happen that wart mannan is going to block both by blocking this. Then PDE is inhibited. So there's just different looks at all this same stuff. What's happening here? Insulin receptor via IRS, insulin receptor substrate, right? That's not like tax revenue service. PKB, and you're looking at PDE and PDE's role, and there's PKA, and see hormone sensitive lipase and perilipin. This is actually a really good diagram, this one. And so these diagrams, these cell signaling cascades, the more you look at them, don't just look at them during lecture, like that's lazy, right? Look at them more, like either review lecture, you can Google these cell signaling cascades, because this section is going to be really cell signaling cascadey. I mean, it is sort of mechanobiology and phosphorylation cascades. I mean, that's really what we're getting into. And hormones tend to work this way. Now, binding proteins, this is IGF-BP1. Obviously, insulin-like growth factor binding protein 1. IGF-BP2, IGF-BP3, that one appears to have a halo around it. This is 80% of all of your IGF is bound to that one. 4, 5, and 6. The liver is making a bunch of these things, and it's making the bulk of your IGF. The liver isn't making all of your IGF, but it's most of your IGF is made in the liver. And as with binding proteins, and about 98% of your IGF is bound. You see like IGF-1 receptor, IGF-2 receptor. IGF-2, this is like you're a fetus and you're developing and stuff. IGF-1 is used. This is the one we're going to talk about. This is IGF, insulin-like growth factor 1. That is what is affecting you as an adult sitting here listening and exercising earlier or later, or whatever. IGF-1, we're going to talk about too. The IGF binding protein, protease, right? It's a little Pac-Man that looks angry. This is like, shark teeth. And so if we're going to lyse, these binding proteins, because IGF, it's not going to bind if it's bound to a binding protein. So again, about 98% of your IGF is bound to binding proteins, and the bulk of it is bound to binding protein 3. Now, the functions of these things, the functions of your binding proteins are in part to extend half-life, right? You're going to extend the half-life, which would be like storage and fight degradation, extend half-life. Those are sort of the same thing. Same with inactive form. It says inactive form. I meant to say inactive form. Modulate hormone activity. This is really the same thing, these two. So when you see lists of what binding proteins do, there's some overlap here. Increased solubility in the blood. Okay, fine. Yeah, we'll give you a solo act for that one. But storage, fight degradation, extend half-life. Let's just lump all those into the same category, because these hormones, the half-lives aren't forever. It's not like, well, I got some leptin, satiety hormone. My fat cells are expanding. They kicked out some leptin. Oh, I don't need to eat for a couple of days. That's not how it works. I mean, it's like half an hour for leptin is the half life of this thing. I mean, testosterone is shorter, insulin's even shorter, epinephrine is just like, a little over a minute. It's very brief. And so if you bind stuff to binding proteins, you can fight that degradation, you can prolong that half life. But then also these bound hormones, if you're bound to your binding protein, you are affecting the activity of them. Okay, let's get into some specific hormones and muscle protein metabolism, anabolism, catabolism. Every single muscle fiber, you remember this stuff. Each one of these is a nucleus. There are lots of nuclei, myonuclei, muscle cell nucleuses, myonuclei. And the cells are so huge. Huge cells need a lot of nuclei for, governments. I mean, if your entire country is the size of like a Rhode Island, fine, one government will suffice. If you have like 50 states, though, you need 50 governments. I mean, every city has its own government, mayors, and so that's the same thing with the larger, these huge muscle fibers. You need a ton of nuclei in here. And now this is a nice little list of anabolic pathways, catabolic pathways. It is not comprehensive, but follistatin, that's a binding protein, right? It binds to myostatin. So follistatin, binding protein that binds to myostatin. IGF-1 is going to work through PKB, AKT or PKB, mTOR. TOR is fine. At some point, I have a totally different name. Mammalian target of rapamycin. The recent literature in the last couple of years, or whatever, 4 years, maybe something like that, is mechanistic target of rapamycin. That's stupid. Just call it mammalian until they stop calling it TOR. Target of rapamycin is a stupid name. TOR, mTOR sounds cool though. It's a great sounding name, like the noise your mouth makes. I mean, it sounds like, I don't know, a good swordsman in Game of Thrones, or it's some sort of like fantasy character here, mTOR, or whatever. It's like, it's a nice name, but what it stands for is sort of stupid. It's a historical name. And then androgens, testosterone, androgens. A lysosome, lysis, soma, right? Loosen the body, organelles with a bunch of the enzymes, these hydrolytic enzymes to break up stuff. So we're not going to talk too much about lysosomes. We are going to talk about the ubiquitin proteasome pathway, little ubiquitin tags, and you degrade these proteins. We'll talk about that. And then myostatin, fine, that little myokine. Follistatin is anabolic because it binds to myostatin, which is catabolic. So there's follistatin, right? It's a glycoprotein, it's a binding protein, and so myostatin, this Belgian blue cows, just if you've never seen these before, Google Belgian blue cow. I did some Googling. There's a Belgian blue dog, essentially, but they're myostatin knockout. Now, myostatin knockout, this just means selective breeding to get glutes like that. So this is some serious glutes. J-Lo has nothing on those cows. So these, or whoever, what was the new one? I don't even know. She's not even new anymore. She's like 100. Whatever. Who cares? Somebody in the comment box. What is like the glute lady of the day? I don't know these people. I'm waiting impatiently. There it is, Kim Kardashian. Yeah, so, Kim Kardashian is not a myostatin knockout. A Belgian blue cow. But look at the musculature here, and so Kimmy K's aren't real. Okay, that's weird. I just find fake things, parts of anatomy that are not anatomy. I don't know. I find that weird. I might be a minority in that position. But so these Belgian blue, that's not a Belgian blue dog, but one of the effects is we're inhibiting PKB, myostatin. You're inhibiting PKB, so you're halting mTOR signaling, which is translation of proteins. Let's get that ribosome to ramp up some protein, to cook up some protein. You inhibit that cascade. You also, prevention of myoblasts from differentiating into mature fibers. For the sake of this class, let's talk about PKB. So if you inhibit PKB, I keep talking about PKB being the most anabolic enzyme in the body, right? And this is how a lot of these hormones are working through of all these enzymes. If myostatin is inhibiting PKB, you're inhibiting that signaling cascade. Now, IGF is turning it on. It's turning on the same signaling cascade that myostatin is turning off. Now, endocrine, the bulk of it, the bulk of your IGF is endocrine, but there's also autocrine and paracrine, and muscle cells are producing this. And MGF, you'll often see it listed as it's produced by fat cells. So stuff in the body is going to produce IGF, but the liver makes most of it, of your IGF-1 and its binding proteins, and that's an endocrine effect. And again, that hypothalamic pituitary liver axis is you go from the hypothalamus, growth hormone releasing hormone. You can also, if you release somatostatin from the hypothalamus, you're inhibiting this, but the release of growth hormone. So growth hormone releasing hormone from the hypothalamus, from the anterior pituitary, right? This is blood flow from the hypothalamus to the pituitary. It gets there by these vascular channels, and then the pituitary, the anterior pituitary, releases its growth hormone, and then eventually, right, you're going to get a bunch of IGF released. It's going to take some time. It's going to take a little bit of time before that IGF gets released, and again, most of your IGF is going to be bound. If you were to just get a bunch of proteolytic enzymes and chew up all of your binding proteins and release all of your IGF, I mean, you die, right? This diabetic coma, because it can bind to the insulin receptor, and you're going to translocate all of your GLUT4 as you get all that GLUT4 up to the surface and just wipe your blood of its sugar. Now, actually, if you're keto adapted, you're probably not going to die. There were some studies done a long time ago. This could not fly today. Nobody would do this. I wonder if I've mentioned these before, though, where they just keto adapt people and they just inject them with a ton of insulin. They have like no blood sugar, and like, oh, I'm okay, and like if they weren't keto adapted, they definitely would have died. That's not an experiment you can run today. You can look it up. It was a long time ago, and I don't know what the values of things were or anything, but it was just sort of a hilarious experiment that is a landmark study because it can never be repeated. But PKB, it's you are activating your PKB. Now, getting into androgens, getting into testosterone, right? That's what's down here. Andro, gen, man, make. So if you talk about like android fat, that's like male fat, that's like stomach fat. Gynoid fat, what does a gynecologist do? It's female fat, right? And so that's like your hip fat. That's your Kim Kardashian fat. Andro means man. It's like android fat, whatever, man fat, like beer gut. So androgens, androgenesis, testosterone is the most potent androgen. And again, they're made from cholesterol, they're fat soluble, they bind to their receptor and get this hormone receptor complex, and that's what has its nucleic effects. Different levels, you don't need to know anything about the levels. Who cares? This isn't physiology, right? This is just like numbers, and so the only thing I really care about is like the actual physiology of things, and not the numbers of like how much testosterone is normal. Like, yeah, who cares? But they're just so there's a range. But then when you look at the range for females, it's way, 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 way lower than it is for males. And younger males, get your sleep. Notice, like in the morning, it tanks. You get to the sort of evening, and for young folks, for young guys, it's sort of the lowest time, and so you have this diurnal, like circadian rhythms, diurnal rhythm, this 24-hour cycle of testosterone. Older guys, it just doesn't really change all that much. For younger guys, there's a diurnal rhythm there. The root word, right? Witness. So you testify. That's what testes mean. And so this is the hypothalamic pituitary gonadal axis. Now, luteinizing hormone, that's what LH stands for, is luteinizing hormone. For guys, that's testosterone production. For women, it's more like ovulation. Tons of effects. Who cares? Tons of effects in this stuff. Depending on your profession. Now, this was a 1990 study. I haven't seen it replicated. Someone's probably done some replication of this, but it tends to go with your I don't know, with confidence and success, and like cockfighters, you just give the roosters a couple of easy wins early on, and they're gonna get more testosterone, and anything you, if you're a sports team, go up against some easy teams at the start of the season, and you're going to develop an endocrine response to it. Now, I don't know how robust that response is. You can go find studies on that, but whether you're like a cockfighting rooster, or you're a baseball team, or whatever it is, go get some. You're like a cage fighter, get some easy wins under your belt. If you go start with some losses, like you're going to be down here in the kind of ministry with your testosterone levels. You get some early wins, right? And you might be like NFL or actor or something like that. Now, 1990, I think firemen were very different. We have published out of our lab some issues with firemen, and it's like a public health concern because they just hang around playing like Halo all day in their like there's just like so many of them are overweight, and their cholesterol is bad. And so more firefighters now, the leading cause of death I think is heart attacks in firefighters. And so maybe, right, maybe they're still up here, but it'd be useful to read it. Like there's me, I'm like tied with like homeless people. I'm tied with like unemployed people. So, but you can affect it just with nutrition. So all you have to do is go on a high fiber diet, especially stuff like beta glucans. It's a type of fiber in oatmeal. It binds to bile, and so you shit out bile, and as opposed to recycling bile, and bile is made from, in parts made from cholesterol, and so you get on this high fiber diet, and a low fat, low cholesterol diet. And what you see is, so this was a study that did the bidirectional study, is more interesting. So baseline to low fat, and then to high fat, baseline to high fat, and then to low fat. So you take a bunch of guys, and ages 50 to 63 years old, and they change their diet. And then what they see is when you go from high fat to low fat, so from baseline you go on the low fat diet, your total cholesterol goes down, right? So total cholesterol goes down, LDL goes down, HDL goes. That's like the good cholesterol, the high density lipoproteins or lipoproteins also goes down. But your total cholesterol goes down a bunch when you go on this high fiber, low fat diet. But what follows, you make testosterone from cholesterol, so your testosterone goes down too. Dihydrotestosterone, androstenedione, dihydroepiandrosterone. When you look at all of these, all of your androgens, those follow, those go down, and they don't fully recover if you go baseline low fat and then back to high fat. Well, let's start here, baseline to high fat. So, total testosterone, it goes up. Just go on a high fat diet, that thing goes up, and then it tanks when you go on the low fat diet. But when you go baseline to low fat, all right, it tanks. Going on a high fat diet doesn't quite recover. So these big phases of super low fat diets, they do have consequences of the witness variety, of the testament variety. Androgens, obviously, I'm talking about. So let's get on to growth hormone. And now growth hormone, it's not just like a thing, right? There are these variants. There's lots of forms of growth hormone, these aggregates of it, and I don't know, blood levels, who cares, whatever. But the pharmacological, human growth hormone, the H is actually important there. It's not just a useless like human growth hormone, because it doesn't, the receptors in a human being, the growth hormone receptors don't cross react with animal sources, right? And so the human growth hormone, it used to be really expensive. I'll talk about that in a minute, because you can't just go shoot an animal and take its pituitary and use that. Use come from cadavers. Not a lot of cadaver growth hormone to go around. So in the 80s and before, it was super expensive. But lots of stuff affects growth hormone. Tons of things affect growth hormone. Sleep, meals are weird. I mean, some people are like, oh, it goes up with meals, but if you go fast, if you just go like deny yourself food for 48 hours, you'll have this big first ghrelin, ghrelin. Remember, that's released from your stomach during times of absence of food. GHR in ghrelin, growth hormone releasing. That's ghrelin. That's it. And so you fast, you just stop eating for a couple of days, right? And your growth hormone goes up. downstream from ghrelin, GHRLin. So food stuff, sort of ignore, I don't know, whatever. But exercise, also, it depends on the nature of exercise, right? Sleep and exercise, wonderful. Now, women tend to have levels that are at least as high as guys here. Guys, testosterone is doing a lot of the tissue interaction. Women, your testosterone levels are way lower, and so you have growth hormone IGF doing a lot of that regulation. So for women, it's a lot more PKB, mTOR types of signaling that way. The types of the characteristics of exercise, get a bunch of lactate going, right? High intensity. And that seems to be long, sort of volume training. That really seems to be what's doing it. So long, intense stuff. Don't go work out for 15 minutes. Don't jog or I mean, just really intense stuff. Change the pH and get a bunch of lactate built up and catecholamines, and that seems to be the big stimulator for growth hormone. And again, hours and hours later, at least 8 hours later, a while later, you're going to see IGF being released from the liver. Now, sleep also, if you're denying yourself sleep, that's going to be problematic. Sleep deprivation. But so the changes in some of these hormones with go exercise, 30 minutes, 60 minutes, 90 minutes, you see cortisol is going to go up and eventually go down, right? And free fatty acids are going to go up. It takes a little bit of time for this stuff to happen. Growth hormone takes time, and then that spikes. Takes time before it spikes. You see like epi, norepi, you work out for a minute. All right, fine. You're going to start getting that stuff. I mean, you might get like anticipatory responses. You're at the starting line and your heart's going crazy or whatever. But growth hormone, this takes a little bit of time before that's going to start going up, and then even longer before your liver decides to release its IGF-1 and the binding protein, right? And then this is a lot of the tissue interaction of growth hormone is actually coming from your IGF. It's not the growth hormone specifically. Somatostatin, right? This is going to somatostatin will inhibit the release of growth hormone. Growth hormone does have its own signaling cascade, JAK-STAT, the JAK-STAT signaling cascade. And so it's Janus kinase and signal transducer and activator of transcription. So transcription and translation. Translation is the creation of protein, RNA. We're on the side of transcription. So it does have its own effects, mechanisms of action. But you give somebody a ton of growth hormone, just have a pituitary, probably, gigantism. If you're Richard Keel, this is, I think, Moonraker. He's Jaws in the James Bond movies, and Happy Gilmore. The guns don't kill people, I do. Whatever, like sort of giant. Your anterior pituitary, a little bit too much growth hormone here. Starts to get, like, look at his forehead. It gets acromegaly. Acromegaly. It sort of makes you get like the Neanderthal forehead. You get this bone development. Or Andre the Giant, even better. Andre the Giant. I mean, that guy with his little tiny teeth, because your teeth don't respond to this. So you just have the little tiny teeth compared to this giant head. And look at him on these. That's two plane seats. I mean, he's sitting on two different plane seats, and it's too small for him. Here's like a regular sized beer can in his hand, and somebody, at some point, you have to Google Andre the Giant beer and then look at his records. And there's some story about it. If you read As You Wish, Carrie Elwes's book about Princess Bride, and he is basically just this homage to Andre the Giant and how wonderful of a human he was, but there's a lot of drinking stories in it. So the anterior pituitary, if this is overexpressing, right, you can be a little bit Andre the Giant-y, but really it's the IGF has a lot of these interactions. So Andre the Giant was billed at 7'4", 550, died at 46, right? You can't be that huge and live to 50. I mean, it's surprising they made it to 46. He was sort of so messed up by the Princess Bride days that that was 1986, I think it was being filmed, something like that. 85, 86, came out 86, 87, somewhere around there. He died in 93, not that much longer. And he was so messed up by then. It was just, he couldn't even lift the princess, right? He couldn't even lift Buttercup. So those shots where he's lifting Buttercup, either he's facing the back and it's someone else, or she's propped up and he's not really carrying her. I mean, it's not like an awesome thing once you're past your prime. Big Show, where he came into the whatever it was, WCW, or yeah, WCW. He was originally billed as Andre's son, but he had his surgery on his pituitary. Now, if you were just remove the whole thing, I mean, he's going to have to take some vasopressin so he doesn't wet the bed, right? If the posterior pituitary is gone too, he doesn't have vasopressin, and so how do you keep pee in? And so there are other functions obviously in a pituitary that there comes with a lifetime of medication, but he's going to outlive unless something goes weird, get rid of that pituitary. Otherwise he'd keep growing and be huger than Andre the Giant eventually, maybe. And but he didn't get quite the same dimensions. But again, before 1986, I mentioned this earlier, it's cadaver pituitaries, and this recombinant HGH was more affordable afterward because you can sort of synthesize it in the lab. On athletes, we don't really have any good studies on using GH on athletes, and so you just have to do sort of an epidemiological perspective. Oh, this person used GH, and here's what happened to them. The liver, this is the hypothalamic pituitary liver axis, and you can detect it in blood tests, and I am unaware if some sort of urinary test came out. I'm unaware of it. Does tons of things, in part because hypothalamic pituitary, and then it just goes out and just has more effects from there. But growth and enhancement of immune system. And so if I ask some question about what are the effects of GH, and it degrades collagen. No, it doesn't. Obviously it's like everything grows, and so that would be like the answer to a question about GH. Now the last one we'll do, I'm pretty sure the last one we'll do, is the hypothalamus. And the again, the HPT axis. So the hypothalamus, that releases the thyrotropin releasing hormone, and then the pituitary, the thyroid stimulating hormone, and then the thyroid. That's when we get to the T3, T4. And T3 is a little bit more potent, but we have the right and left thyroid glands. Now, it's if you are facing, it's like you're right and you're left. It's not if you're looking at the person. So obviously, like this is the left side, but we're facing the person, and so it's theirs. And so Dexter is the right side, and Sinister is the left gland. So the right thyroid gland is called Dexter, and the left is called Sinister. That's from the coat of arms. That's just what it's called on a coat of arms. Chief and base if we had two more. So it's not like it's some horror movie or something. Triiodothyronine and thyroxine, right? T3, T4. This one's more potent, but this is mostly what you're releasing. And hyperthyroidism, too much, right? Hypothyroidism, your metabolism is you're freezing, and your metabolism is bad. Now the goiters, I didn't take this picture, but I've seen some pretty huge ones in Cameroon when I did this little medical mission in Cameroon, and there's some mighty goiters. You just need iodine, right? You just need a bunch of iodine, and in a Western diet you get it, but you get this hyperplasia of the thyroid cells. And we've understood how this thing works in part for quite a while, and I love like the very German discussion with the K. But 1965, thyroid hormones and regulation of protein synthesis. This isn't a new investigation, but new investigations continue to come up. And in part, how the thyroid is working is PI3K, PKB, mTOR, P70S6K, right? So this is the mTOR signaling. This is cell signaling. This is like cell surface. This is how IGF, and insulin, and stuff like that is activating protein translation. But so it has genomic as well as classic cell signaling, what you would expect from a protein, from an amine hormone, what you would expect. It is also enhancing cell signaling cascades. And aerobic exercise, right? If you're doing a bunch of days of training of aerobic exercise, you can get this sort of hypogonadal effect of sort of exercise-induced decrease in testosterone with too much aerobics. Dave Hooper, he did a study recently. I'll go dig it up, and maybe I'll put it in one of the slideshows coming up. about the hypogonadal male of just go jog all the time, your testosterone goes down. So you can do this by food, right? You can do it just mess up your sleep and be very like defeated in life, and eat a low fat, high fiber diet. There's lots of things can lower testosterone. And again, you don't want tons of testosterone. If your testosterone is too high, like goodbye heart, and so there's or relationships with cancer. If you have too high of testosterone and there's a tumor in there, that's growing too. So all things in balance, but just consider goals, whatever your goals are. Okay, what questions do we have? Are we on the no question day because it's Friday and we're ready to start a weekend?
I actually had a quick question about beta 3 receptors.
Okay.
Is that the mechanism we see through beta oxidation through for lipolysis?
No. Beta oxidation is you're chopping up the free fatty acid itself. And so, lipolysis, the beta threes, that's the cell surface receptor, um, for on a lipid, right? And so you're going to go through lipolysis, through that um, beta adrenergic signal. So the beta oxidation is all right. Now we have these fatty acids, right? We have these carbon chains. Let's just say it's like 16 carbons or something. And let's go chop that thing up and make some acetyl-CoAs, right? So if you have 16 carbons in it, well, let's go make 8 acetyl-CoAs, right? And so you have to get into the mitochondria first, a mitochondria. And so we transports like carnitine, and you could usher it into the mitochondria, and then you start chopping off little, what becomes acetyl-CoAs. So that's the beta oxidation, is the consumption that follows lipolysis. You can't beta oxidize a huge triglyceride. You can't have this like a huge molecule and like, all right, burn this, right? That doesn't work. So you separate it into its little component parts, right? 3 fatty acids that are now free fatty acids, right? And then these acyl CoAs go. Each one of those little strings goes and gets chopped up. So that's the beta oxidation part comes next, which I'm not going to talk about in this class because it is um, it's not as much like muscle phys. The balance between pKa and pKb sort of is the heart of muscle phys, of the balance of what pKa is doing and what pKb is doing is your muscle metabolism. And are you growing? Are you shrinking? Is it hypertrophy? Is it atrophy? And that balance. But so I don't really care too much about the lipolysis itself, other than pKa signaling. That's what pKa signaling is doing. And so to understand pKa signaling of let's break up all of our stuff, it gives us a picture of what pKb is doing. If you can't do pKb without doing pKa first, at least I would prefer not to do pKb without having first done pKa.